All right, Ninja Nerds, so what we're going to do in this video is we're going to talk about antibody structure and function. All right, so antibodies. There's actually these ones that we have right here, 1, 2, 3, 4, 5 antibodies that we have listed, right? What is an easy way? There was an acronym that I found from someone who actually helped me when I first took this anatomy and physiology, was this little mnemonic here called GAMED. And GAMED gives you all the antibodies that you'd have to know. IgG, IgA, IgM, IgE, IgD. So it's a quick little way of being able to remember all the 5 antibodies. All right, so now what we're going to do is we're going to go into detail into each different type of antibody. So let's go ahead and dive right away to the IgG antibody. So IgG antibodies, one thing you're definitely going to want to know about this guy, he is the most abundant antibody. So out of all of these antibodies, he is the most abundant antibody, okay? So a good portion of him, literally about 65% of him, 65 to 70% of him is actually found within our blood plasma. So 65 to about 70% is found within the blood plasma. And again, who secretes these antibodies? We already know this. The plasma cells, right? So our plasma cells, if you remember them, the one with that really big nucleus and, again, a large amount of rough endoplasmic reticulum, these are the guys that are responsible for producing this one. In this case, what is this one right here called? This is IgG antibodies. Now, IgG antibodies, we're going to get into its structure in a little bit here, but this guy is actually designed to be what's called a monomer. What is a monomer? That means he just has one of these antibodies here. So there's only one of them. So what is this guy here again? He is actually a monomer. All right. So IgG antibodies are monomers. So there's only one of these puppies, all right? Now, we know plasma cells make these guys, right? We know that it's the most abundant antibody. We know that 65 to 70% of it's found within the blood plasma. This antibody is actually produced within it's produced within both the primary and the secondary, but it's the most abundant one in the secondary immune response. So it's more abundant, or the primary one, in the secondary immune response, but it is produced in small amounts in the primary immune response. So in a primary, meaning that you first get infected, secondary is that you're getting exposed to that antigen or foreign pathogen again. And we'll talk about that when we talk about the graph. And we'll talk about somatic hypermutations. All right, so again, he's producing the primary and the secondary immune response. Okay, next thing. What does this guy do? So if you remember from before, you'll remember that we had a bacteria here, right? And if you remember that bacteria, what can be on the cell membrane of that bacteria? Or the cell wall, right? Depending upon what type of bacteria it is, gram-negative or gram-positive. You know that it can have those antigens, right? So here's that antigen. Let's actually draw this antigen in blue here. So here's this antigen, right? And then what can be bound to that antigen? We can have the IgG antibodies bind to that. And if the IgG antibodies bind to that, what can that do? That can activate, if we remember already, it can activate the complement system. And what can the complement system do? That can initiate the formation of a membrane attack complex, or they can enhance opsonization, right? So that's one thing. They can form the membrane attack complex, or they can enhance the opsonization, right? And they can cause C3 and C5a, which enhance inflammation. That's one mechanism. Another thing they can do, let's say that there's a virus particle. Here's a virus. So here's a viral molecule here. So it's got like this nucleocapsid, and then it's got some, so let's say it's RNA or DNA that's actually present within inside of it. And then let's say it has like these specific types of viral antigens out here on the surface. What can happen is those IgG antibodies can literally bind to every one of these antigens. And whenever it binds to these guys, this virus can no longer come and bind to an actual host cell. So it can't infect it. So what is that called? That's called neutralization. Neutralization. Let's say there's a free antigen, right? So there's some type of free antigen circulating in this vicinity of the plasma or within the extracellular fluid, right? And what happens? These antibodies, they bind to this free antigen. and precipitates this antigen, which can enhance phagocytosis. So this can do two things. It can actually initiate opsonization, and it can initiate phagocytosis by trying to enhance the phagocytosis. But what is this process right here called? This is called precipitation, right? So it can do precipitation. What else can it do? It also, because it can do precipitation, it can do neutralization, it could activate our complement proteins, and there's a lot of other things that it can do also, right? One other thing I want to mention is, you know that whenever a baby is within the in the gestational phase, right? So it's actually in the fetus stage, and there's the placenta that's developed around the 12th week, right? Well, the placenta, let's just say here's our placenta right here. The placenta can actually provide, so here let's say some blood vessels coming from the placenta, right? They're like the umbilical veins. They can allow for these antibodies to circulate through. So let's say here I have IgG antibodies. These IgG antibodies can actually pass over from the placenta where? To the baby, right? And so let's say here's your baby, right? So here's your baby. All right. What can it do? It can pass these IgG antibodies onto the baby so that that baby has what's called passive immunity. So whenever it's born, it's already going to have a small amount of immunity, right, to specific pathogens that the mother's been exposed to. So that's kind of cool. So again, what do we have for the IgG antibody? Most abundant antibody. 65 to 70% of it's found within the blood plasma or extracellular fluids. It's secreted by plasma cells in the monomeric form. It's secreted within the primary and the secondary immune response, but it's more abundant within the secondary immune response. And what is its effects? It can enhance the complement proteins by doing the membrane attack complex and opsonization. It can bind and cause neutralization of certain types of viruses or other microbes. It can cause precipitation reactions, which can enhance opsonization and phagocytosis. And it can also pass the placenta and confer passive immunity between the mother and the baby, right? That's that part for IgG antibodies. Now, let's go to IgA antibodies. So IgA antibodies. All right, so IgA antibodies, you would find these. Where would you find these? Because we talked about these guys. Where would you find these guys? And again, well, first off, who secretes them? Again, it's going to be the same cell constantly over and over and over again, but we're going to keep mentioning that so you don't forget it. This is actually going to be the plasma cells. So the plasma cells secrete this antibody. But IgA antibody is interesting. Look at this. You notice something right here? Look at that. They're linked together, but it's not a monomer. There's two of them. So if there's two of them, what do we call this? This is called a dimer. This is called a dimer. So again, plasma cells secrete these IgA antibodies, but they're in the dimeric form. So it's two monomers linked together. So this is a dimer. All right, so now, where would you find these IgA antibodies? Well, these plasma cells, they're secreting these antibodies that's present within a lot of the actual fluids within our body. For example, you can find this within the saliva. You can find this within the skin, so it can be on the skin secretions, like your sweat secretions, so we'll put sweat. You can find it within the mucosal lining of our GI tract. So the mucosal lining of the GI tract. Oh, and one really, really good one too. This is an important one. I'm going to mention this one. Within the milk, the milk of the mother, right? So whenever the milk, so we'll call this the milk produced by the actual mother within her alveolar glands, right? So the milk from lactation. So whenever the baby is actually suckling and it's actually gaining that milk from the mother, these IgA antibodies can actually get pushed into that actual milk. And those IgA antibodies can get passed from mother to the baby. And that can help with some of the actual immunity. That's why you want to be able to breastfeed, right? All right, so again, that's pretty much these ones that you would find them in. Again, so it's a lot of our actual body fluids like saliva and the sweat and the mucosal lining of our GI tract and within the milk and even with some within the urogenital tract and stuff, right? So we can even say urogenital tract. But now, what are these designed to do? So what they love to do is they love to be able to destroy certain types of bacteria that might actually show up within this area. That's pretty much it. How can they do that? They can do a lot of the similar mechanisms that we already talked about, right? So again, these IgA antibodies, they're really, really good at being able to, let's say we have a bacteria here that somehow gets inside of the actual GI tract or within the saliva, or it's actually on the skin, or it's within the mucosal lining or whatever. Then you would have, let's say here's the antigen here, you would have this IgA antibody here that is actually going to be a dimer, and he can actually bind this point right here. But if he's actually a dimer, maybe there might be another bacteria within the vicinity or some type of microbe right here. And what can it do? It can also bind to that antigen. So again, that's why he's a very good one right there. He is a dimer. All right, so that's pretty much about IgA antibodies. Again, you'd find it within the saliva, the skin, the mucosal lining, certain types of milk within the mother, and you can even find it within the urogenital tract. And just know that it is a dimer. Okay? Next one, IgM antibodies. So IgM antibodies are actually going to be another very, very important one. And again, we should continue to keep doing this. Who secretes this guy? Plasma cells, right? So the plasma cells are the primary cells that are going to be the only cells, really. There is other cells that can make antibodies, but these are going to be the primary ones. And these guys are going to be our plasma cells, and they are secreting IgM antibodies. You guys are going to crap your huggies right here, because this guy, he is a pentamer. He can, look at this guy. He can exist in two forms. He can exist in a pentameric form, and he can exist in a monomeric form, right? So we have two of these guys right here. We can exist in this form, which is what? 1, 2, 3, 4, 5. This is a pentamer. And it's all linked together through what's called a J protein. But again, this is a pentamer. So it can secrete these antibodies in the pentameric form, or they can be secreted in the monomeric form, right? So there is two different forms of this antibody, the monomer and the pentamer. IgM antibodies are very, very important, right? So they're secreted by plasma cells, and they're pretty abundant too. Now, what do these guys do? So these guys are the ones that are secreted within your primary immune response. So this is the one that's made during your primary immune response. All right, so this is important. Why is this important? Because this could be a good diagnostic tool. If you actually run somebody's actual blood or their antibody titers and you find out that they have elevated levels of IgM, it could actually let you know that they're undergoing some type of active infection, some type of primary exposure. First time they're being exposed to some type of foreign antigen, and they're producing antibodies against that, right? So that's important for understanding that these IgM antibodies are made during their first primary immune response, right? What can these antibodies do? All right, well, these antibodies, and they can also be found within a lot of our extracellular fluids also. This guy, he actually is going to love to be able to activate the complement protein. So if you remember here, we had that bacteria. Let's say here we have that bacteria. And if we have that bacteria there, and again, what did that bacteria have on its cell membrane? It had specific types of antigens. And what can happen is this monomeric form of the antibody can bind on to that guy right there. And then what can happen? We already know what's going to happen. You could activate the complement proteins, right? And the complement proteins could lead to membrane attack complex or opsonization, right? So that's that. What else can he do? He's really good at being able to pick up. So you know whenever you get a mismatched blood transfusion? If you get a mismatched blood transfusion, so let's say that you have a mismatched blood transfusion. So here's your red blood cells here. Let's say I have a couple red blood cells here. And these red blood cells here, they all have foreign antigens. Maybe it's an A antigen and you weren't supposed to give an A antigen to this individual, but here's this A antigen, and you have, let's say, anti-A antibodies. Well, guess what? This IgM can actually bind right here. Look at this. This is a beautiful thing. And then all of these guys can bind here through, again, what is that molecule called? It's called a J protein that links them all together. This right here, this molecule right here is our IgM antibody. And what is he doing? Remember when we talked about this, in the blood typing videos, that whenever we have any type of mismatched blood transfusions and there's a antibody-antigen interaction, well, an antibody-antigen interaction can lead to what's called agglutination. And agglutination is clumping, right? And that can cause a lot of problems here. So again, there can be agglutination. This is a type 2 hypersensitivity. All right, so this is actually what's called a type 2 hypersensitivity whenever there is a mismatched blood transfusion. And we'll talk about that more in pathology. So again, what can this guy do? Okay, he can be made by plasma cells in the pentameric form or the monomeric form. He can be made during the primary immune response, and he can even be made a little bit during the secondary immune response, but very, very little. What does he do? He can bind on in the monomeric form to foreign antigens and initiate the complement system and activate the membrane attack complex and opsonization mechanisms. Or he can bind on to multiple different antigen binding sites. So he has multiple antigen binding sites. If you think about it, when we draw the structure of the antibodies and discuss them, he has 10 different antigen binding sites or epitopes. So he has 10 antigen binding sites. And for right now, you're just going to have to trust me what I'm saying, that it has 10, because you might be like, oh, it only seems like it has 5. Well, you'll see here in a second whenever we talk about it. Okay, so again, that pretty much covers IgM antibodies. Let's go to the next one, which is IgE antibodies. All right, so now IgE antibodies. What do IgE antibodies, where are they secreted from? Again, let's just keep doing this just so we can be consistent here. They're made by plasma cells. But we can kind of expand on this a little bit. So they're made by plasma cells, all right? And when they're made by these plasma cells, so they can be secreted by plasma cells, because these are the only ones that are really releasing the antibodies, the plasma cells. Plasma cells are the only cells that are making these antibodies. Now, IgE antibodies are monomers also. So these are also monomers. So these are monomeric proteins or glycoproteins. So this is a monomer. And where would you actually find this guy? Well, you can find it within the actual respiratory tract. So the respiratory tract mucosa. You can also, so it's found within the respiratory tract mucosa, and you can even find it within some of the urogenital structures too, right? So you can also find it within the urogenital structures. And you can also, we're going to discuss another thing, you can also find it within the lamina propria too, or certain types of lymphatic tissue. So we'll put lamina propria or and lymphatic tissues. Okay, so now, these antibodies are pretty cool. What do they do? All right, so let's say we talk about the respiratory tract mucosa first, because this has a very important role within what's called type 1 hypersensitivities or anaphylactic shock. You might have heard of that, right? So let's say here, I have a mast cell. You guys remember mast cells, right? So mast cells right here, we talked about this in other videos, but we didn't go into super depth on it. You know how you have these receptors, right? These receptors are called, on the mast cell, Fc epsilon R1 receptors. So again, what is this called? Fc, which stands for the Fc portion of the antibody, epsilon R1, okay? So this is the Fc epsilon R1 receptor. Why do I mention that? Well, guess what? You see this monomeric antibody? Whenever there's some type of inflammatory response, that monomer will literally bind in right here. So that monomer will literally bind in right here. So now you have the Fc epsilon R1 receptor bound with the monomeric IgE antibody. Why is this important? Because whenever someone is exposed to some type of allergen, something that they're allergic to, for example, let's just say it's a ragweed pollen that I draw here in blue. Whenever these ragweed pollen molecules activate this actual IgE antibodies, it can signal the actual mast cell to start producing tons of different types of inflammatory chemicals. So again, you already know these molecules. What are these molecules called? They're called histamines. And I'm not going to go over all of them, but they could be leukotrienes, right? So they could be leukotrienes. They could be prostaglandins, right? So, and what are these molecules designed to do? Well, if you remember, they cause widespread vasodilation, right? And another thing that they're going to do is, there's also going to be other molecules that are produced too. Whenever they cause that widespread vasodilation, it's going to cause the blood vessels to start getting leaky. And if this is happening within the respiratory mucosa, if you imagine here, if I were to draw someone's bronchi here, let's say I draw a bronchi, and I have a blood vessel right here, serving that bronchi. And I have a lot of capillary permeability coming out of that. I have an increased capillary permeability. What's going to happen to that actual space right there? It's going to start getting bigger and bigger and bigger. And it's going to cause a lot of edema. And that's going to create swelling, which can actually start restricting the airways. All right, so that's why people can actually have a problem breathing. So there would be a lack of oxygen coming into this area here and a lack of CO2 getting out. And that's because of that, a lot of that edema here, that accumulation of fluid there. Another thing that happens is these actual molecules, they bind onto different receptors. Now we know that they cause vasodilation, right? They also bind onto smooth muscle cells here within the bronchi. And when they bind onto these smooth muscle cells within the bronchi, they actually cause these smooth muscle cells to actually contract. And when the smooth muscle cells contract here, so it's going to cause contraction of the smooth muscle cells within the bronchi, it actually decreases the size of the airway. And if you decrease the size of the airway, can as much oxygen get in and as much CO2 get out? No. And that can affect breathing, right? That's what people know to be called as anaphylaxis, right? So anaphylaxis. This is actually a type 1 hypersensitivity, okay? That's one of the big important things with these respiratory tract mucosa is that they play a very huge role within these allergic reactions, specifically type 1 hypersensitivities or anaphylactic shock, right? All right, what's the other thing? Urogenital it's also important of, right? And it's also important within these lamina propria and lymphatic tissue. It's also important within the GI tract mucosa. Why is it important within the GI tract mucosa? Let's say that I decide to eat some type of uncooked bear meat and I'm an idiot. I don't know why I would ever do that, but I did. So here's my GI tract, right? I eat this undesirably uncooked meat or whatever it might be, and I end up getting, somehow I get parasites. So I get a parasitic worm in here. Look at this puppy right here. I got a parasitic worm floating around in me. That's nasty, right? But what happens? These actual parasitic worms, we actually can produce antibodies against them. And guess what antibody we produce against them? IgE antibodies. And then we can tag these IgE antibodies for what type of white blood cells. You should know this. This is actually going to be our eosinophils. So if you remember from before, what was this actual cell called? This was called a eosinophil because he loves to release the major basic protein and the cationic peptide, which cause destruction of the actual parasite. So it'll come over here and it'll actually destroy that parasite. All right. So that's that for the IgE antibody. And there is small traces of it. You can find tiny amount of traces of it within the blood plasma. So very small traces in the blood plasma. Okay? So that's a cool antibody there, all right? Now, let's come over to the last and the final one, the easiest one, IgD antibodies. IgD antibodies are, again, they're made by plasma cells. But you're going to love what they do because they're not that hard to remember. They're actually made by the plasma cells, and they are also monomeric. So these are also monomeric, all right? So they're only going to be one of these molecules here. So this is a monomer. But what do you think this guy does? It's actually not bad. Guess what? He decides to come back, or actually, he wouldn't be secreted because you know proteins, whenever they come from the Golgi apparatus, they can actually become a part of the cell membrane or they can be secreted. I just wanted to show it so you guys could see this guy here in a larger view here. He actually is going to, whenever the Golgi apparatus has those vesicles, it's actually going to fuse with the cell membrane. And when it fuses with the cell membrane, it's actually going to express that actual monomer on the B cell surface. So this is our B cell receptor. So this is going to act as a B cell receptor. What we talked about in the actual humoral immunity or the adaptive immunity video, right? So he's a very good B cell receptor. All right? So that pretty much covers everything for him for the IgD antibodies, right? So he's a very good B cell receptor, just like the actual monomeric form of the IgM antibody. All right? So again, come back over here to the IgM antibody really quickly. Another thing that can actually happen with this monomer that I didn't mention is that this monomer can also exist as a B cell receptor. So he can also exist as a B cell receptor, just as the IgD antibody can. All right? Now that we've done that, I want to talk about what's called somatic hypermutation. What is meant by somatic hypermutation? All right, so if you remember, we talked a little bit about this already when we talked about the IgM and the IgG antibodies, how they're a part of the primary and the secondary immune response. So here on the y-axis, I have the abundance of antibodies within our serum. And here on the x-axis, I have the time this is actually going to occur over time in days. So let's say, all right, first thing, I get exposed to an antigen. Let's say I get exposed to antigen X. So I get exposed to antigen X. What happens is, after we're exposed to antigen X, there's a lag period. So there's this lag period where nothing's happening, right? So here's this lag period. But then let's say after maybe a day or something like that, we start producing antibodies. And this starts rising. Look at this, it starts rising. All right? And then after that, it'll actually come back down. But then there's another antibody that'll also be produced, but he's actually going to be produced a little bit later, even that, longer than that. Look at this guy here. And then he comes back down. So whenever we have our first exposure to an antigen, we produced an antibody a little bit, there's a lag period. What is this thing right here called? This is a lag period. And this lag period is actually going to take some time and for our plasma cells to start producing antibodies. Let's just say for this case it's 2 days, whatever. Let's just say it obviously could even be a little bit longer, maybe 5 days. Let's say 2 to 5 days, so 2 to 5 days. And then what happens? During this lag period, we start producing a specific type of antibody. What is that antibody? This is called IgE antibody. That's the first antibody that we're going to produce during the primary immune response. But then later on in that primary immune response, we make another antibody. And this antibody is actually going to be called, and as this antibody is going down, this antibody is actually going up. That's interesting, right? Because now that means, what does that mean? That means our plasma cells were designing themselves to secrete what at this point in time? IgE antibodies. But then over a certain amount of time, they switched. And then they started producing IgG antibodies. Well, how does that happen? That's weird, right? The genes get activated by certain types of interleukins and cytokines. So there might be, for example, for this IgM antibody, there might be certain types of cytokines here. So there might be certain types of cytokines or stimuli, like the microbe itself, that's activating this guy to produce IgM antibodies. Whereas there might be other types of cytokines, or there might be other types of microbes that is actually going to tell this plasma cell to start changing its gene sequence, changing the actual genetic transcripts, and producing IgG antibodies. So whenever there's this change or this shift from IgM to IgG antibodies, that change in the production, this right here is called somatic hypermutation. Okay, now, we're not done yet. So now, as this starts dropping, and then eventually this starts dropping. So again, what is this antibody here? This is IgG. Here's where somatic hypermutation takes a definite, huge change. So again, let's say we're coming over here. It's that we're not actually being exposed to anything. And then right away, I give a second exposure. So now I'm exposing them to a different type of antigen. Maybe I'm just having a second exposure to X. If there's a second exposure to X, now watch this. The IgM antibody actually has a little lag period, but then it produces it again, but it's not as much. Now watch what's going to happen to this black line here. It's going to go ham, and it's going to go all the way up here, right? So there's that change, right? So look how much of a difference there is here. This is the big difference. This is really the definition of, this is really the better example of somatic hypermutation, because primarily we were making IgM antibodies, right? That was the first one that we were making. And we still do make a tiny little bit amount of him in the secondary immune response, but look which one we're making primarily, IgG antibodies. So this is how we actually should describe somatic hypermutation, is that how we were making IgM antibodies primarily and IgG at almost equal amounts, pretty much close to equal amounts. There was a greater lag period for the IgG though. And then afterwards, when we were exposed to the antigen a second time, we didn't even make that many IgM antibodies at all. We primarily produced IgG antibodies. That is somatic hypermutation. Okay, now, we're getting close to done. Let's talk about a couple more things and then we're done with this lecture here, right? Immunity, there's two types. One type is actually going to be called passive, and the other type is actually called active. Now, what is the difference between passive immunity and active immunity? Passive immunity means you didn't have to do anything to be able to get that form of immunity. It was given to you without you having to do any work, all right? Or produce any antibodies in response to it. So you were given these. It's like someone actually, me working to be able to get money, or me just getting money from somebody. If I'm just getting money because I'm an heir, it's a passive form, right? If I'm actually working to gain that money, that's active. That's how this is actually going to work. So let's explain what I mean by this. So passive, there's two types of passive immunity, as there is two types of active immunity. Passive, there's what's called naturally acquired. So there's the ways that you can naturally acquire immunity, and then there's a way that you can artificially acquire immunity. All right, so artificially acquire. What are these ways that you do that? So naturally acquired. We already talked about one example. It's a perfect example. That would be the IgG antibodies and the IgA antibodies. So again, what is these? IgG antibodies and the IgA antibodies that we got from our mother, right? So IgG antibodies, that would be the ones that cross the placenta. And then the IgA antibodies are going to be the ones that we got from the mother's milk. These are naturally acquired. We didn't have to do anything to get these. We were given these naturally, right? So if we were given these molecules, if we were given these IgA and IgG molecules naturally, we didn't have to work for them. They were given to us, right? All right. Artificially acquired. This one would be, let's say that I got bitten by a snake. I got bit by a snake, and the snake has a specific type of venom, right? And that venom is actually causing damage to my tissue, and I have to get the antidote. So then what they do is they give me an artificial form of the antidote, which is an immunoglobulin. So let's say that they give me an anti-venom, or maybe I got bit by a black widow, which has a latrotoxin. They gave me specifically antibodies, immunoglobulins, that will actually ward off that venom. So it's an anti-venom. So you can think about that. I didn't do anything against it. I didn't produce the antibodies. They gave me the antibodies. So think about that as like a anti-venom. The anti-venom is actually immunoglobulins. And what do I mean by immunoglobulins? I'm just now mentioning this, and I'm sorry. This is another word for antibodies. So antibodies, they literally gave me antibodies against the actual, so these antibodies are against the venom from a snake. And this could be other examples. This is just the first example that popped in my head, right? So that's artificially acquired. Now let's come over to active. Active is the one that we actually have to produce antibodies against. And again, there's two types. There's naturally acquired, right? Naturally acquired. And then there's also artificially. All right, so again, naturally acquired, what does this mean? So naturally acquired is I was exposed to it. So me actually being infected with E. coli. So me actually being infected by pathogen. Because if I'm infected by a pathogen, like maybe Streptococcus B hemolyticus Lancefield type A, strep throat, right? I have to produce antibodies against that pathogen. That is an active form of immunity, naturally, all right? What is the artificial form? This would be an example of vaccines and booster shots, right? So vaccines and booster shots. Why is this active? Because you know in vaccines or in certain types of booster shots, they literally give you a week. So let's say here's the antigen. This antigen is weak, or we can even say dead. So they're weak or attenuated or they're dead antigens. And then what happens is my immune system produces antibodies against that actual weak or dead pathogen. And then when I produce antibodies against that weak or dead pathogen, that is an artificial way of having active immunity. And that could help us to be able to have specific types of immunity against certain types of flus or viruses or certain types of bacteria, right? That's the purpose of artificial vaccination. All right, last thing and then we're done, guys. Let's talk about the structure of an antibody. Let's take the structure of our antibody. So what we have here is we actually have these two different types of chains here. Look at this. So right here, I have what's called this whole part right here, all the way up, all the way up, all the way up, right here. I'm going to shade this in right now. This whole thing right here is called our constant heavy chain. And if we have one here, we should have one right here too. So all this right here is also constant heavy chain. Then we have another chunk of it over here. Let's draw that other chunk over to here. So now we have two chunks over here also. So we have a constant heavy chain. Then over here, we got another piece right here. This piece right here that I'm going to shade in, and this piece right here that I'm going to shade in. This is our constant light chain. And this over here is our constant light chain. This right here, and I'm going to draw a little bit of a different structure here. It can actually kind of look like this maybe. This is our variable heavy chain. And then this right here, this can also have a different type of structure here. This is our variable light chain. And the same thing over here. This would be variable light chain. This would be variable heavy chain. And then look what we have connecting these guys together. Right here, we're going to have disulfide bonds linking that guy together, disulfide bonds linking this part together, and disulfide bonds linking that guy together. This is the structure of the antibody. Why is this important? All right, let's finish up here. This portion of the antibody is a good attachment point for complement proteins. Complement proteins love to be able to bind to these portions right here. All right, that's one thing. Second thing that's really, really important. You see how I drew these variable regions a little bit different? These variable regions differ from antibody to antibody. In other words, let's say right here, I have 3 different types of antigens. This undergoes recombination in such a way that every variable region is different for every type of antigen. So in other words, if this one is going to be for this one, right, it's going to make a variable region that's specific to this antigen right here. If we wanted it specific to this antigen, what would it take on? The variable shape would look like this. And if we wanted to be able to fit this, what would it look like? It would look like this. That is the purpose of these variable heavy chains and these variable light chains, is that they can undergo recombination to make different structures to accommodate these different types of antigens, which makes them good for being able to bind on to foreign invaders, right? Plus, if you think about it, I have 2 binding sites here for one antibody. So this technically has 2 antigen binding sites. What does that mean? Remember when I told you I'd explain to you what was the purpose of that antibody for IgM? Let's come over here real quick. Remember I told you it has 10 antigen binding sites? It's because of that. Each one of these antibodies has 2 antigen binding sites, and it's a pentamer. That's a total of 10 antigen binding sites. All right, guys. So in this video, we covered a lot on antibody structure and function, all right? All right. I hope you guys enjoyed it. I hope it helps. See you, Ninja Nerds.